Assalamu alaikum girls. Today we are going to discuss mitosis in today's lecture. So please refer to figure number 5.2 for clear understanding of the whole process. So whenever I am explaining, you please keep an eye on the diagram, okay, and all these phases. It is not important and it is not mandatory to use this diagram for the exam purposes. You can make it separately also, okay, like this. It starts from here, like for example you are starting from, so you make this prophase separately, then this, like, prophase, these two diagrams, one, then second down, and then you can make it horizontally and you can make it vertically. Any kind of any kind of arrangement is accepted, but it should be correct, okay. It should not be in the form of this octahedral form or any other form. You can make it vertically, you can make it horizontally, but the order should be correct and the figures should be correct and the labeling must be correct, and the name of the phases must be correct, please, because all of you, like most of the students, they are making these mistakes, okay. Okay, so in 1880s, a German biologist whose name was Walther Fleming, the name was Walther Fleming, he observed that in a dividing cell, the nucleus passes through a series of changes which are known as mitosis. So it is a type of cell division in which the cell divides into two daughter cells. Please, you have to differentiate between meiosis and mitosis. Please keep your concepts correct, okay? Okay, so you have to differentiate between the mitosis and the meiosis. You must know the process completely and without errors, and it should be there without any confusion. It should be very clear in your mind, okay. So what is mitosis? It occurs in eukaryotic cells, so uh, and in multicellular organisms. uh, now it like it divides the cell into two daughter cells, each with the same number of chromosomes and it as they were present in the parent cell. So the main phenomena is that the cells divide. One cell gives rise to two cells. Okay, so one parent cell is giving rise to two daughter cells, and with the same number of chromosomes, which were, like, see, the genetic material is the chromosome. Okay, so the nuclear material is same, but there are two now. Okay, for example, okay, so I'm giving you an example here, that, see, for example, if this is yours, if this is any kind of um a thing, like, it's, for instance, it's just a living thing, it's just imaginary, okay, it's imaginary, and it has two small like chromosomes in it. Just imagine they are this is a nuclear matter. So when this cell divides into two, what happens to the nuclear matter? This also divides, understand? So one goes over here and the other goes over here, yes or no? So now these both newly formed organisms, which are the daughter cells, they have the nuclear material but in half, understand? This is not the nature's principle. The nature's principle is to divide the nuclear material, to divide the nuclear material uniformly. If this is a nuclear material, if this is the nuclear material, then it has to be complete here and then complete here also, understand? For example, mitosis takes place in the area of your body which is wounded. Okay, if you have any kind of if there is a cut, or if any kind of wound is there, it is healed with the help of mitosis. So it has to make the same cells. For example, you have a paper cut, and then after some days it's healed. How? Because the cells are taking place, because the cells are growing and the cells are taking place of the other cells, and your wound is getting healed. Understand? How is the healing process taking place? Because new cells are being formed, and if the new cells are different from the parent cell, how will they take place of the older cells? For example, if the DNA is different, how will they become a part of the skin cells? Because now there are different skin cells, because the nuclear material is half. After the division, again the nuclear material is half. At a point in time, what will happen, it's just imagine, okay, that the nuclear material will vanish, because it is getting half, and then it's becoming half of the half, then half of the half. So this is not how it works. Okay, the nuclear material has to be constant, it has to be uniform, it has to be equal in each and every cell. Newer cell, older cell, the nuclear material has to be same in your, for example, the cells in your hair, the cells in your nails, the cells in your skin, all have the nuclear material which is same, equal, and the number of chromosomes is same also, which we are going to study. Okay, now see, somatic cells are those cells which form the body of organisms, while, see, this is very 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 important point. What are somatic cells? Somatic cells are those cells which form the body of the organisms. For example, each and every cell of your body is a somatic cell, but except those cells which, while the germ-line cells, now the germ-line cells are those cells which give rise to gametes. So somatic cells and gametes, these are two important things. Somatic cells and gametes. Gametes, what are gametes? That we will be studying later, I think. Somatic cells undergo mitosis, while germ-line cells undergo meiosis. Yes, uh, we will be studying gametes in meiosis. Okay, so now just think about somatic cells. Oh, actually, the gametes are the reproductive cells. So the reproductive cells have meiosis. The meiosis is related to the gametes, and mitosis is related to the somatic cells. Each and every cell except your reproductive cells, except the reproductive cells of the each and every organism, like I'm talking about the eukaryotes. Okay, the eukaryotes, they and the multicellular organisms, they have mitosis in each and every cell of their body except the gametes. Okay, now phases of mitosis. What happens is, the process of mitosis is complex and highly regulated. Highly regulated means um, it has a proper discipline and a proper order, so it's regulated. Okay, there are two major phases, that one is the karyokinesis and other is the cytokinesis. So what happens during the karyokinesis is that the division of nucleus is divided into four phases. Actually, the karyokinesis is the division of the nucleus, and the cytokinesis is the division of the cytoplasm. So karyokinesis and cytokinesis, you must know the meaning of these. Now we are discussing the karyokinesis. Okay, not the cytokinesis. Cytokinesis is a process which happens after the karyokinesis, and it is like an easy process. The main is the karyokinesis. Cytokinesis is very easy to understand, and it's a simple process. Okay, normally the genetic material is the nucleus is a loose thread-like form which is known as chromatin. So how does the chromatin look like? Chromatin looks like this. Okay, and at the onset of prophase, like when the prophase starts, chromatin condenses into highly ordered structures known as chromosomes. You already know the structures of the chromosomes. Now this becomes into this highly structured chromosomes. Okay, and then and they have a centromere here. So in the center they have a centromere, and this is the center of the chromosome, which is known as the centromere. And now what happens is that the genetic material has been duplicated in the S phase. Remember, in the S phase I said that the um, genetic material has been duplicated. Okay, now it means that each chromosome has two sister chromatin which I made. Okay, now each chromosome also has a kinetochore at the centromere. Kinetochore is a complex protein structure that is the point where spindle fibers attach. Okay, So now you will be seeing what are spindle fibers, how do they attach, what is a kinetochore. Actually, there are two centrioles collectively called as centrosome close to the nucleus. Remember, two centrioles which are known as centrosome and which are present close to the nucleus. What is the function? Now, these each and every centriole, it does what? It duplicates. And what happens? Two daughter centrioles are formed. Both the centrosome. Now we are talking about the centrosome. Okay, please don't confuse the centromere with the centrosome. Centrosome is related to the centrioles, and the centromere and the um, chromatid is related to the chromosome. Okay, now they give rise to microtubules by joining the tubulin proteins. Actually, what is happening is the spindle fibers are going to the spindle fibers are constructed. Okay, complete set of spindle fiber is known as mitotic spindle. By this time, the nucleolus and the nuclear envelope have degraded. The spindle fibers have invaded the central space. Now, actually, what happens is that spindle fibers are formed, and they are known as mitotic spindle, and the nucleolus and the nuclear envelope, they degrade. Why they degrade? Because they have to see, the nucleus the nuclear envelope has to degrade, and the nucleolus also has to degrade. Why? Because the cell is going to divide. So we are the nucleus has to degrade, and because the spindle fibers will spread and the chromosomes will align themselves. Okay, in highly vacuolated plants, the nucleus has to now we are talking about plants. First we were talking about animals, now plants. uh, in highly vacuolated plant cells, nucleus has to migrate to the center of the cell before prophase. The cells of plants lack centrioles. Now did not have centrioles. Also, the spindle fibers are formed by the aggregation of the tubulin proteins. Remember the tubulin protein on the surface of the nuclear envelope during prophase. Okay, so they uh, do not have they do not have the spindle fibers. Uh, Actually, they do not have centrioles. So the spindle fibers are made with the help of tubulin proteins. Okay. Okay, one more very important point is mentioned here, that the prokaryotes do not have a proper nucleus. See, as we all know that the prokaryotes, for example amoeba, for example euglena, for example paramecium, they also have to divide like this. For example, this is one amoeba and it has to divide. So what happens in an amoeba, that it divides into two. Okay, this is one amoeba and this is the another one. So how it is going to divide? This process is known as division, but it is not known as mitosis. Why? Because no spindle formation is here. I will be discussing the spindle formation with you. So this is just normal division, and we can call it reproduction, but it is not mitosis. Why? Because there are no spindle fibers involved. What are spindle fibers? Now we'll be discussing this. Okay, so see, here the cycle starts from the prophase. This is your prophase, again prophase, and this one is also prophase. So today we are going to discuss still here. Okay, this prophase, and this prophase. Okay, we are not discussing the other phases, the metaphase, anaphase, telophase, cytokinesis. No, nothing. We are not discussing it. Okay, so what happens is that the chromat. This is now see. This is the centromere. So this is the centromere, and this is the blue point is the kinetochore. So if you see clearly, you can see the kinetochore also in each and every chromosome here. Okay, first of all, see, the chromatin here condenses and the centrosome replicates. Where is the centrosome? This is the centrosome. it is going to replicate, okay, and the chromatin is getting condensed. This chromatin is getting condensed. Condensed means it is going to get visible now, and this will become in the form of chromosome, okay. See, in the S phase, the nuclear material has been doubled already. But now what is happening in the process of mitosis? What is happening is that the chromatin is condensing and the centrosome is replicating. Okay. Now this, see, now they are now condensation is continued and the spindles are formed. What are spindles? These pink threads are spindles. Okay. Now what happens is that, see, where is the nuclear envelope? This is the nuclear envelope. This is the nuclear envelope which is going to break. Now what happens is that this nuclear envelope is broken and it results in this kind of an arrangement. So if you see clearly, the kinetochores are getting attached. Where are the kinetochores? See, this is a kinetochore, and again this is, here is a kinetochore. So this is how the kinetochores are here. Two kinetochores, this one blue and again this blue. These kinetochores are attaching the chromosome on the spindle fiber. Understand? So if first to give you an overview, see, this is, see, this is, so we are going to start the overview for the revision. And please, you must, please girls, you must know that any kind of answers which are not from the book, even if they are from any kind of key book, guide, anything, they are not acceptable. The answer must be from the book. Understand? I am telling you, the answers must be from the book, nowhere from any guide or any kind of key book. No answers from there, from them are acceptable. Okay. So this is the, this is the centrosome which is replicating. This is the chromatin, and this centrosome is replicating and it is becoming into it is turning into spindle fibers, and then these are condensing, and this condensation, this condensation makes the chromosome appear, okay, and then now they are more clear. Second is that the nuclear envelope is dissolved and it's broken, and then what happens, they align themselves into the form of the in on the spindles. From where? From the kinetochores. Understand? Please, if you don't understand anything, ask me.